Hello and welcome to Lesson 5, Localization of Brain Function. In this session we are going to look at the research that has investigated how different brain functions are localized to specific areas of the brain. Some key questions are going to be: what is the function of the Wernicke's and Broca's area? What is the difference between the somatosensory and motor cortex? And how discretely separated are different regions in the brain? So let's start with a couple of definitions. First of all, what do we mean by localization? Well, this refers to the idea that specific brain functions such as language, vision, etc. are located in specific geographical regions in the brain. Lateralization is something we'll be going on to at the end of this presentation and continuing next time. Refers to the idea that a hemisphere is specialized for certain function, so that specific functions are dominated in one hemisphere rather than being equally represented in both. So let's have a little look at the research. Franz Gall in early 1800s was a pioneer of sorts. He developed the theory that personality was linked to bumps found on the skull. This became the field of phrenology, and he was one of the first really to identify the idea that personality might be localized to specific regions, in his case of the skull or the brain. And his research, although laughed at today, perhaps laughed is a bit harsh, but rejected today, actually has a germ of truth in it. Cesare Lombroso also had a similar idea that criminality could be identified by the atavistic form or certain physical features. If you want to have a look, I have a phrenology head on my desk. Feel free to have a handle and look. So in 1861, Paul Broca was the first researcher to identify a specific area of the brain and a discrete function that it seemed to be responsible for. What is now famously referred to as the Broca's area. He noted that patients struggled to produce speech if a specific area of the frontal lobe, this frontal lobe here, was damaged. This area is now called the Broca's area. Wernicke in 1874 discovered that difficulties in speech comprehension was associated with damage to a part of the temporal lobe. So, speech production and speech comprehension. This seemed to indicate that cognitive functioning was highly localized. Just a quick recap to the brain: the frontal lobe is this blue area here, the parietal, the temporal and the occipital. Remember, occipital is largely to do with vision, auditory processing is here, some higher functioning here, and the parietal lobe we'll talk about perhaps in a moment. So, localization of brain function. There are many functions, cognitive functions, and there have been many areas that have been suggested as the localized processing area. So we have Broca's area for speaking, we have the motor cortex, which we'll look at in a bit more detail, for movement, sensory cortex for sensation, the visual cortex in the occipital lobe, the Wernicke's area, and the auditory cortex in the temporal lobe. Each of these have slightly more specific functions, and there's some stuff here about how we can actually measure them. So we're going to look at EEGs in a few lessons time, PET scans and postmortems and so forth. The brain can also be stimulated by using magnets, and one way we can measure functioning is by stimulating them using transcranial magnetic stimulation. I told you about my experience on transcranial direct current stimulation. If you can't remember what that is, I'll talk to you again in a lesson about it. So let's look at a specific area of the brain and function: the visual and auditory cortex. Now, information from the retina is sent to the visual cortex. Damage to the visual cortex can cause blindness. However, other areas located near the visual cortex are responsible for perception. Being able to see is not the same as perceiving something. Okay, so that's quite a complex idea. So you could stop the signal from reaching at your visual cortex. You could stop the signal. You will have seen the thing, but you will not have perceived the object. Damage to the secondary area. So damage to the primary area, the visual cortex, results in blindness, but damage to these peripheral areas, these secondary areas, can lead to perceptual abnormalities such as prosopagnosia, which is can't recognize faces. The man who mistook his wife for a hat suffers from this. We get a chromatopsia, where you can't see color, and visual field impairment, which we're going to look at a lot in Sperry's research later. And this is usually occurs as a result of stroke. It's literally where part of your visual field is just invisible to you. So that's one area, the visual and auditory cortex. Another area of the brain referred to as the somatosensory cortex is responsible for sensation and perceiving sensation. Somatosensory, soma, remember, means body, so somasensory means body sense. Essentially, information is received from receptors in the skin. These include pressure, pain, touch, and temperature. Body areas then are mapped within the somatosensory cortex, and you can see a slice through here which shows how different functions or different areas of the body are represented. So legs, hips, trunks, neck, head, upper arm, elbow, moving down the body, teeth, tongue, and abdominal organs. So essentially, if I took your eye and poked it, this part of the brain would be active, and if I pinch your forearm, this part of the brain would show activity. So it's very, very localized and very systematically organized. This is the homunculus, which is a way of depicting how many sensory receptors, or how sensitive those receptors are to processing. So we have many, many receptors in our hand which allow for very finite detection and movement, and the same goes for our tongue and our lips and our mouth and our nose and our eyes, but we have relatively few in our abdomen and our ribs and our calves and so forth. So that's the somatosensory cortex. It's this sliver here, and this represents the different areas. The motor cortex is similar to the somatosensory. It's laid out very similar. Muscles, legs and feet are at the top, and then moving down to very complex mouth and tongue at the bottom. Now if you think about the types of movement involved in tongue movement, they are substantially more complex than knee movement, which is antagonistic muscles. So these are laid out in a very similar way to the somatosensory cortex. Carl Lashley, 1920, carried out some really interesting research with rats. He was interested in learning, and he made lesions in the rats' brains and observed how long it took them to solve maze problems and remember mazes. Lesions made to the visual cortex resulted in impaired maze learning. Experimented with different sizes of lesions. Small lesions he found had little impairment. So he argued this. He came up with two laws. The first was learning impairment was dependent on lesion size. So learning spanned multiple regions. So he's really criticizing this idea that very discrete areas of the brain are solely responsible for specific functions. He says no, it's multiple areas that are responsible, and that's why these larger lesions are the ones that damage the rat's ability to do this, to solve the mazes, because they span multiple regions. So maze solving, or memory, requires multiple regions. And then the law of equipotentiality, and this is different areas have the same capacity to be used for learning. Here we have a very early proposal of neuroplasticity, We're going to do a whole lesson on this in a few more time in future. But this is the idea that any area of the brain could take over the function of any other. Here is the rat's brains and an example of the different types of lesions that he carried out. This was revolutionary. So that's brain localization. You know, some evidence that certain functions are localized, Broca's, Wernicke's, whatever it may be, visual, occipital. Some evidence that multiple regions are actually responsible for the same function. Another element to brain organization then is what we call hemispheric lateralization. So we have two hemispheres, the left and the right. Somatosensory and motor processes are organized and span both hemispheres. So we saw that sort of useful little slice through the hemisphere there. As a result, somatosensory and motor processes are crossed. So the left hemisphere controls the right side of the body and the right hemisphere the left. So they go sort of opposites. Now, that's interesting because I have a stroke characterized by damage to the right hemisphere. This will paralyze essentially my left arm and the left side of my body. You can see it in this image here where the field of vision from the left part of the eye actually is processed in the left hemisphere, but the right field of vision is transferred over to the right. Both hemispheres mirror each other's functions, so even though if we have a functional structure here, we'll have a corresponding functional structure over this side as well. So, Paul Broca investigated a patient called Tan. Tan could understand speech but could not produce anything. The only word he could say was tan, which is where he gets the name. When he died, Broca performed an autopsy, a post-mortem examination, and found lesions towards the base of the frontal lobe. He investigated others with similar speech programs. He found lesions in the brains that were very similar. However, these lesions were only ever found in the left hemisphere. Now, lateralization argues that every structure in one side of the brain may have corresponding structure in the other. However, Broca's research disagrees with that. It says that actually language is processed mostly, at least speech production, mostly in the left hemisphere. Reading, writing and talking. So this produced an early model of speech and language. The idea would be something like this. Information is sent from Wernicke's comprehension to Broca's area where you explain your comprehension. Research has shown that reading begins in the visual cortex and onto the angular gyrus, a really important structure. This is then passed on to the Wernicke's for comprehension. So you see the word, interpreting the word, understanding the word, and then if you're copying it down, you may be producing the word. Damage to the angular gyrus is rare but results in inability to read. You don't like reading, you don't have damage to the angular gyrus, just for the record. You just don't like reading. Limitations of Broca's work then. So the idea that brains are lateralized or they may not be. Let's evaluate Broca's research. Dronkers et al. Looked at brains preserved from two of Broca's patients, found extensive damage to other regions as well. So it's difficult to actually, if the independent variable was damage to the Broca's region, the frontal lobe, we have all these confounding variables because there's damage to other regions as well. Research has shown that lesions that affect only the Broca's area generally only result in temporary speech disruption. This suggests language is distributed function, it is a distributed function and not entirely localized. Communication versus localization. Desjardins described a case in which a patient lost the ability to read as a result of damage to neurons connecting the visual cortex to the Wernicke's area. This suggests that reading which is a complex behaviour, relies on the functioning of a whole network, not just one single region being damaged. And then neuroplasticity and individual differences. Evidence from plasticity research shows that Lashley was largely correct, that function can be recovered by utilising a different region of the brain. Harris-Day et al. 1997 found that language associated cortical regions are proportionately larger in the female brain, and this probably explains gender differences. Women use language much more than men. One point, just go back to the neuroplasticity though, one limitation of this is that neuroplasticity seems to be limited to age. So after about 27 years, the plasticity of your brain has reduced dramatically. A couple of exam style questions then. Pair up the definition with the brain region and discuss localization function in human brain. This is going to require a lot more research from outside of this topic, such as plasticity and the evaluations that I provided you with. So that is brain localization and lateralization. As always, if you have any questions, please do not hesitate. Otherwise, thank you very much for your time. I will see you around. Bye now.